All right, and welcome again, everybody, to the first review session for the Anato-Bee. We have a lot of really good practice questions all prepared for you in anatomy, histology, and embryology. So we're really excited to help you guys get a little bit more confidence in terms of what you're going to expect for the Anato-Bee competition. So with that, we realize we also haven't introduced ourselves in a while, and you may have joined us a little bit later in the tutoring sessions. And so we thought it'd be good to start with that. So I'll let Dr. Haggerty give us a go and let us know who she is first.
Hi, my name is Dr. Jenna Haggerty. I am an assistant professor at Thomas Jefferson University, specifically at the Cindy Kimmel Medical College, where I teach medical students, PAs, third and fourth year medical students, histology, anatomy, neuroanatomy, all of those fun topics that you guys are actually going through and learning yourselves for this exam. I am also one of the co-founders of the Anato-Bee with Dr. Piscira and Peterson. And I did actually, fun tidbit, used to work with Dr. Peterson at Drexel. And then I am actually really good friends with Dr. Piscira. So that's fun. And a nice, fun little individual note for myself, personal note, is that I have two cats. Their names are Sir Wesley Fluffernutter and Miss Princess Buttercup. And then I have twin boys. I just had twin boys in August. So it's nice to meet all of you, and I'm excited to go through this review with you today.
Thanks, Dr. Haggerty. I'll let Dr. Peterson take over now.
Hi, everyone. My name is Dana Peterson. I am a professor at the Drexel University College of Medicine. Both myself and Dr. Frank, we are on the West Reading campus, which is about an hour west of Philadelphia. So we're about an hour west of where Dr. Haggerty teaches. Both myself and Dr. Frank consider ourselves to be jack of all trades, and I will say personally for myself, a master of none. But I have been teaching these different content areas, all those that Dr. Haggerty just mentioned, for more than 30 years. So I'm looking forward to tonight's session, and I hope all of you find it really, really helpful.
Dr. Frank, if you could give us some information about yourself as well.
Well, as Dr. Peterson said, I work at Drexel University College of Medicine, and I work with her in the anatomy, as anatomy faculty at the West Reading campus. So I have 3 children and a lovely wife, Miriam. I have no cats. I have no dogs. I'm a prior cat and a dog owner, but currently unaffiliated. And like I said in my little bio, that if I'm not teaching or doing something with the anatomy lab, I like to either listen to or play music. And I say from Mozart to Megadeth. So more than you wanted to know, but you asked. So good luck to everyone. And I hope you enjoy and find this review very helpful.
Thanks, Dr. Frank. And finally, I'm Dr. Mary Piscira. I'm at the Edward Via College of Osteopathic Medicine in Auburn, Alabama. I only just very recently graduated with my PhD, but I've been loving pretty much everything about anatomy for many years now, and kind of just started where you guys were with just a curiosity. So I've got 2 dogs, and I'm loving how much sunshine we get down in the South, originally from more northern in Ohio and Pennsylvania areas. So really glad to be here and really glad that all of you are here. So with that, I just wanted to remind everybody about the emails that went out. Obviously, if you're here, you were receiving communications from the Anato-Bee to get the link for this tutoring session today. There was another email that went out that was letting you know about what's going on for the local competition and the general schedule for that. So that is going to vary depending on what time zone you're in. So that information can be accessed additionally in the news tab on the Anato-Bee website. But you should be receiving a communication from your local chapter coordinator by this week, if you haven't already. And that should contain the Zoom link for your participation on Saturday's competition this Saturday. Very exciting. So if you haven't received that, or if maybe you know a classmate that hasn't received that, please reach out to us. There might be some issue with the form of communication with the email specifically that you've provided us. So we'd like to fix that and make sure that you're good to go there. So really looking forward to that. Just a reminder about in-person proctors. So we were requiring in-person proctors for the competition, just to secure your testing space, make sure that everybody's keeping their academic integrity. And so just a reminder that if you had a teacher that was playing a role as your in-person proctor, reminder that you'll need to coordinate with them about probably a time to go to your school and have them proctor the exam. Or if your teacher wasn't expecting to do that, find a family member, someone over the age of 18 that can provide that for you. Finally, we just wanted to also say that we have a research survey going out. This in no way, shape, or form affects your participation in the Anato-Bee or the scores that will come out of it. It's completely voluntary, just something if you're interested in helping us learn more about your motivations in the anatomical sciences, then we'd be glad to have your input there. So go ahead and check those out. And again, reach out to us at info@anatomy.org if you had not received any of that information. As always, you can put comments in the chat, but this is going to be a more interactive session today than I think what we even usually do. So feel free to type that in. We'll try to address questions in real time instead of waiting for them all the way at the end. So with that, I will let Dr. Haggerty take over for our first practice question session.
Awesome. I am going to share my screen. All right, can everybody see my screen? Yep, okay. So like Dr. Piscura said, this is going to be a review session. So what that means is I am not going to be covering all the nitty-gritty details like you have been receiving in the previous weeks. I'm actually going to be giving you questions, and we're going to be putting up a polling system. So when I go to a multiple choice question, I'll ask Dr. Frank, can you put up a poll? You'll see a poll pop up on your screen, and then you can go ahead and pick the answer choice that you think is correct. We'll give a little bit of time for this, and then when we think we get most responses, we'll share those results, and then we'll go over those results, and then we'll move on to the next question. For ones that are not multiple choice, I'll just say, does anybody have any idea about this? And if you do, or you want to answer, feel free to put it in the chat. That's the easiest way to do this versus having anybody unmic themselves and say the answer. So that's kind of how this will go. So I hope you enjoy playing along. All right. So for our lecture objectives, you're just going to go to the website and you're going to take a look at the lecture objectives. There's way too many for me to include on this screen here. So we're going to start with our first question here. So what structure is indicated in the image? On the left, you can see that it's indicated by the yellow dashed line, and on the right, I'm indicating with arrows. But what structure do you see in this image here on the right? Dr. Frank, if you could put up a poll for us. There you go. So you should be able to see your poll, and when you think you have an answer, getting a lot of good answers. And I think I'm going to wait up until about 50% participation. You guys are participating really great. And I'm just going to go ahead and I'm going to end the poll myself now. And I'm going to share our results. It looks like a majority of you did answer A, which my mouse here is the correct answer. So what we're looking at is the basement membrane here. We did have two other choices that were popularly chosen, which was the dense regular connective tissue and the keratinized stratified squamous epithelium. So that's kind of understandable that I would think these two would be chosen. Okay, so because what we're looking at here, and this is why we're practicing this, because a lot of you haven't taken exams like this. And basically what I'm showing you here is similar to how I'm going to be indicating, or how we say tagging on your exam. Okay, so the indication is this demarcation here by the dotted line. So where my dotted line is here, what structure is that? Or we look on the right, we're looking at, I'm pointing to this area here with these arrows. That's why I'm using multiple arrows. So that's why basement membrane is the correct choice. Superficial to that is the keratinized stratified squamous epithelium that we see here. And deep or inferior to that is the dense regular or really irregular connective tissue shown here. Regular connective tissue is another tissue type, but we have dense irregular connective tissue shown below here. So again, I think this is a really good practice in tagging. So now we're going to try our guess at another type of question. So you're going to have imaging types of questions. And you are going to have imaging types of questions, and then you're going to have multiple choice questions like this without an image. So what type of epithelium is found in the epidermis, which is the top layer of skin, and its primary role is to protect against external factors? Again, you're using that poll that Dr. Frank put up for us. Awesome. We're getting a lot of responses in here. Okay, great. I know we're kind of going through this a little quickly, but I'm going to end the poll and share our results. And the reason I'm going through quickly is because we got a lot to review with you guys today. So I think this is great. A lot of you is answering A, which is 100% correct. If you were kind of listening to what I was saying previously, I'm using that same image because this is an image from our skin that we see, and I've blown up the image to take a look at that epithelium, which is closest to our surface, and we can see that skin is going to be keratinized stratified squamous epithelium. And we want that keratinized layer because it's going to add that extra protection, right? And then we know if I gave you this image here to tell what kind of epithelium I'm looking at, we usually look at our top surface layer here to see what the structure is. And I'm seeing these, or cell type is, and I'm seeing these flat egg-like shaped cells. And then I can see there's multiple layers of it. So that's multiple layers of stratified, flat, squamous. And then at the very top, I look to see, is it keratinized, is it not? If I have these thin kind of wispy, like acellular, so no cells in these layers, that's keratinized that I see here. So that's how we know it's keratinized stratified squamous epithelium. Ooh. In my slide here, this is straight from your tutoring session. So this is just a kind of a reminder of where am I getting this information? So one of those learning objectives is to know kind of the functional differences and the structural differences of the different types of epithelium. So we did just take a look at that stratified squamous epithelium with the keratinized layer on top. Okay, so here's our next question. Which connective tissue is found in ligaments and tendons and is densely packed in an organized manner? I do want to say this, the amount of participation and how quickly you guys are participating is amazing. Okay, so I'm going to end the poll here. I'm going to share our results. So it looks like we have a majority of you saying C, which is 100% correct, which is very cool. So dense regular connective tissue. So this is just recognizing where we're kind of finding these tissue types. So dense regular connective tissue is found in ligaments and tendons. And then we have it's kind of how I think of kind of like my cousin here, right? So we have dense regular connective tissue, dense irregular connective tissue. And I would think these two would be very hard to get confused just because of the naming in general, but also because of kind of some, thinking structurally, how could I tell these two apart? So we have our dense regular connective tissue. And we can see, I always think it looks like bacon. So because of the waviness, and you can kind of see the nuclei on the outer areas of it. Whereas in the dense irregular connective tissue, which we commonly find deep to our epithelium, it's more of like a meshwork of collagen fibers, whereas this over here is a more organized fashion of collagen fibers. So very good. Okay, so we're going to take a look at this type of tissue here. So I'm asking you which area indicates the Haversian canal. Okay, So your brain should automatically ask yourself first, what kind of tissue am I looking at? Where would I find the Haversian canal? Awesome. Look at that. You guys are great. You all are rock stars. All right, I'm going to end the poll and share our results. Okay, so a lot of you are answering D, and that is correct. And I see that I have a lot of other people saying A, and I think that is a very good, I could see why you would want to answer A, because we can see these lamellar-like ossifications, right? Those are what usually surround our osteons, which is shown in answer choice C, osteon, Haversian canal. They sometimes call this the central canal. You'll sometimes hear those words used interchangeably. By looking at this tissue, you should know you're looking at bone. But going back to A here, this is somewhat similar in the sense that this is actually a connecting canal. So those Haversian canals within an osteon, if we think the osteon is kind of like a tubular structure, the Haversian canal sits in the center of it. This canal here, this is a Volkmann canal, and I think of it as like, to remind myself, it's a little highway that connects between a Haversian system and Haversian system so that they connect its contents, which is arteries, neurovasculature, right? And I always think of it, it's like a Volkmann canal. It's a little driveway for my little VW Volkswagen to drive through. So that's how I kind of remember that. How do I tell the difference between this and this on this image? Your Haversian will be central in that osteon, whereas if you see this one, it's a little elongated, and that's because you're basically cutting, right? So we have two tubes that we just cut this way through, but now the one that's going perpendicular, we just cut that in half and we can see the whole kind of like I said, driveway going across between those two Haversian systems. And then the other choices here, this B is actually is pointing to the opening here for where my chondrocyte or my osteocyte sits. And then this here is actually pointing, or the dotted line is looking at that concentric circle, the lamella, again, those ossification layers coming out from the Haversian system. So then you can use the chat for this one, mixing it up a little bit. But the question would be, what are we looking at? Are we looking at compact or are we looking at spongy bone? And then, like I said, just type it in your chat and hit enter. Compact, yeah. Perfect. Compact bone. Okay, when we see these circular-like structures, your brain should automatically think compact bone versus our spongy bone. Again, this is taken directly from those tutoring sessions. So compact, the strongest, most dense, providing support. Remember, I talked about that little driveway for our Volkswagen canal, or the VW buggy. Here's an example of that, which will connect those Haversian systems here. And this is just comparing or showing it to the spongy bone, which is shown on the right side here. It's not as organized. You see more of these kind of like, see what I want to call these, like trabeculae kind of like patterns here. Let's go to our next question. All right, so our next multiple choice question, which of these choices lists the appropriate order of the histological layers of the ovary from the outermost to the innermost? Or you can think of it as most superficial of the organ to deep within the organ. This one might take a little bit of what we call mental gymnastics. All right, still, like, you guys are just participating so fast. I love it. So eager, so great. All right, so B, again, you guys are hitting these out of the park. I think that's really great. So you should be thinking first our germinal epithelium, tunica albuginea, and then cortex, and then medulla. So if we go to your slide that you had in your tutoring sessions, and I thought they did a really great job here. We're starting with that outer simple squamous epithelial layer here. And anytime you have like an organ or anything, it's usually has an outer covering. You hear us say things like adventitia, which is more like connective tissue, or serosa, but especially serosa, it's usually that single layer, simple squamous epithelium. So that's what we're seeing here, that germinal epithelium, the tunica albuginea, deep to that, or going towards our innermost layers, followed by our cortex layer, and then our inner medulla layer. So very good. Ah, Dr. Haggerty. Okay, well, I got my, we're not all perfect here. So since I highlighted the answer for you, so let's take a look here. All right, let's do another one. Where if I would indicate this structure right here, and you can you clear the poll, Dr. Frank? A way to do that. There we go. Okay, okay. So if I would indicate this structure right here, so ignore my markings in the question, but if I would indicate this structure here, what is this structure? Can we indicate on the poll what this structure is here? The options are still the same, but what's this structure that I'm indicating with my mouse? Right here. What's that structure? All right. And the poll here. Yep. So a lot of you getting the answer correct. So I see that M line was, or D was the answer you guys were all choosing, and that is the correct answer. Some of you did pick the Z line. So Z line is basically the demarcation between sarcomere to sarcomere, right? So that really, like, from this dark line to this dark line, that's a sarcomere here. Here's another sarcomere here. The M line, the center line right here, it doesn't stain as darkly as the Z line. That's how I tell when I look at these images and these EMs. And then the answer that I was looking for here was the I band. So you should think that this is mainly my thin filaments that are shown here, whereas the thicker filaments, which will show up darker on these kind of images, are going to be in this area here, which is known as the H band. And sometimes it's kind of hard to, where they kind of like twine is the A band, but that's kind of hard to tell the A band between the H band from what I've seen, only because you have to be able to get a muscle that is like, to really compare contracting and then also relaxing, to kind of look at that change in the thin and thick filaments crossing from one another. And then I do have that this is our slide from your tutoring session, just kind of going through, as you can see, those A bands and H bands on this slide or image particularly are hard to tell between. Yeah, and I did see somebody say, this is going by really quickly. I apologize for that. Again, I just want to make sure I'm covering as much as I can for you guys. So I would say if you're having trouble keeping up, just kind of watch, answer it in your head if you don't want to answer directly on the poll. And you can move the poll itself out of the way if you take it and drag it out of the way to see the question. And of course, these are recorded, so you can go back and watch. All right. So now we got another image question. So which statement best describes the structure indicated by the arrow? Make sure you're reading these question choices. And this is the other thing with your exams. Take your time, take a breath, read it carefully. All right. I think we got a good amount of answers coming in. All right. I'm going to ask a question and I want you guys to put it in the chat. Is this nervous tissue or is this muscle tissue? Yes, it's nervous. Okay. So a lot of you had answered, about 45% had answered A, endomysium covering a muscle fiber. Okay, so if you come to an answer like this, you don't even have to read endomysium. If you see muscle fiber and you know this is nervous tissue, you know it's not A. Okay, so the best answer for this, and your mind was in the right track, and this is how I was as a student. I would see endo and go, oh, that's it, that's the, and I take the first one that I see. Read your answers carefully, read the question carefully, read your answers carefully. So we know it's an axon, right? And it looks like the second majority, the most who answered this question did answer B, which is the correct answer. We know it's nervous tissue. So what part of the tissue am I pointing to? We're obviously talking about the different layers, right? The tissue itself, is it an axon? What's covering the axon? What covers the nerve fascicles? What covers the nerve itself? And we're actually looking at these individual little axons or nerve fibers and the covering of them right here in between, which is that endoneurium. And we can actually see over the fascicle would be all of them grouped together here. The perineurium shown here, and then covering the whole nerve itself, which stains a little bit darker, would be over here, which is the epineurium. And then I did include in the next slide. So this is the slide. I added a little bit of notes in here from the person who gave this lecture, but endo means within. This kind of helps if you kind of know where the words are coming from. Peri means about or around, right? So peri being around the fascicle itself. Endo, which is covering the little axons within. And then the epi, which is above or over or on, will be covering the whole nerve itself. And then when you're looking at the other words like the endoneurium, endomysium, neuro means nerve, mesium or myo means muscle. So you can always look for that as well in the questions.
Dr. Haggerty, could you just go back to that question slide? I think it would be really helpful if you took your cursor and showed them the purple dot representing the cytoplasm of the axon, then surrounding that lipid material, explain what that is, and then maybe remind them what a skeletal muscle might look like in cross-section, where the nuclei, where the dark dots would be if it was skeletal muscle.
Yeah, so we have, and that first one, you mean these dark dots here? Is that what you were saying? Sorry, I just can't recall the first thing that you said.
Just where the actual cytoplasm of the axon is, right there in the center of each of those circles.
Right here. And then you can see the white, which should be, so this is a, I believe this is taken from a sciatic nerve, right? So this is a peripheral nerve. So what helps these electrical impulses go through our axons? Do we remember? Can we answer in the chat? What covers that? Myelin, yes. So what cell helps or produces this myelin in a peripheral nerve? You can answer in the chat. Two types of cells. Schwann cell. Oh, you guys are rock stars. I love this so much. Awesome. Yep. So we can see our myelin here. Cytoplasm of the cell directly in the center. Yep. And then, like we said, that connective tissue covering the endoneurium that's covering the axon itself. Did I cover all of that, Dr. Peterson? The muscle, the muscle tissue, sorry. Dr. Haggerty's brain is not the same since she's had children. You'll probably hear that the rest of your life from your moms and parent friends, but the brain doesn't work the same. So thank you for the reminder, Dr. Peterson. So muscle tissue. So we're thinking like skeletal muscle. That's the primary one that I think we use when we're going through the different muscle layers. Keep in mind your skeletal muscle, the nuclei are kind of pushed out towards the edges a little bit. And so it does kind of like come up with a kind of a circular appearance, but you'll see the nuclei kind of and multiple nuclei smushed around those outer edges. Whereas in here, it's all white, right? That myelin comes up very white in this type of stain. And then you have this centralized cytoplasmic structure that is right into the center. So I think those are the best ways to kind of look and tell the difference between the two. Thank you, Dr. Peterson. All right, let's keep rolling. So which layer of a blood vessel contains a simple squamous epithelium called the endothelium? So again, just thinking about endo, epi, peri. Where are these words telling me I'm at? Look at this. You guys are great. Okay. Yeah, 100%. So we are looking at the tunica intima. So we are looking at the most inner portion. This is a vein, a very large, actually, I think on the slide I took this from, they call it a medium vein. If you get real in particular into these types of histology, you have large veins, medium veins, small veins, and their corresponding arteries. But we have a vein here and an artery. And I'm talking about the intima because it has that, when you hear endothelium, it's basically we're thinking the innermost part. So encircling the lumen, which is the white space we see here. And again, that simple squamous epithelium, one layer of those flat cells, which are a little difficult to see. But what I wanted to kind of show here is you can see all the little darker nuclei here, and then here as well. And then I just wanted to show that you have a vein and we have our artery. And the way we can tell easily the two differences between here is that we have a very organized muscular layers kind of throughout our artery here. And so clicker happy, I'm so sorry. And then not as thick in our vein, but I always love looking at histology and comparing it to actual anatomy, because this is one example I can give to my students of how you can see things in the lab. And I'm just going to mention this because I think it's pretty cool, but my students always ask me, how do you can tell a vein from an artery? And when I'm looking at this image here, and even when I'm in the lab, when you touch a vein in the lab, it feels like a deflated balloon. Okay. And when I look at these two images, which one's my deflated balloon? This one right here. And then if I go in the lab and I feel an artery, your arteries they'll bounce right back up at you. Right. So I can tell if I can squish this guy right here, I know it's going to bounce right back up because it's nice and inflated. So that's just a fun fact for you, all of you. So again, arteries and veins. So arteries have those thicker walls, which we did see in the previous image here. And veins are a little more thin-walled. We have a little bit more information, but what I really liked about this slide was just kind of going through and showing you that tunica externa versus the media versus the intima in an artery and in a vein. And as you can see with the intima here, we do have that endothelial layer that we just pointed out. Okay. In which image are we looking at? Which one shows our conducting airway? Is it A or B? All right. I know this is going quick for you, for some of you, but just bear with us. Keep in mind it is recorded. You can go back and watch. Okay, so A, 100%. Great job. So just in the chat, just put one thing that told you that that was a conducting airway. Hyaline cartilage, love it. Uniformity. All right, I'll go with that. I like that. I like that. Okay. It's not crazy. Yeah, okay, I see what you're saying. Yeah, this kind of looks like it's a little scattered and all over the place. I like it. Yeah, the alveoli. So real quick, in our image B, which is you can consider it a respiratory airway because that's where gas exchange occurs. Conducting is just the transfer of air or oxygen into our system or carbon dioxide out of our system. B, if you see that like one layer disorganization, sometimes you can kind of see like acinar or grape-like structures, you're in the respiratory airways. We want a thin strip of different types of stuff, right, to cause the gas exchange to occur. We don't want anything thick-walled like this. That's going to make it difficult. And then in the conducting airways, yeah, hyaline cartilage, that's a big one because trachea, it's got the hyaline cartilage in it. You can also see your pseudostratified columnar epithelium here. You can see there's different layers, right? So that hyaline cartilage is stuck in that muscularis layer here. We have the thicker adventitial layer, most superficial. Very good, guys. I think this is my last one. Okay, so which area over here on the right, an area I mean indicated by these bars, they have a letter associated with them. So which area is shown in the image of the ilium is which layer is the muscularis layer? Okay. Very good. So B, remember there is a circular and a longitudinal layer in our gut tube, right? We have a epithelial layer. This is kind of do your mental gymnastics and really kind of look at the image because the epithelial layer is facing upside down. Sometimes if we're nice, we like to put the images right side up, but that's not always the case. And the epithelium is in our mucosal layer. C would be your mucosal layer. You have your muscle layer here, or the muscularis layer containing our smooth muscle, and then you have the serosal layer shown here. And again, this is taking out of your slide from your tutoring session, and this is just to show those layers and to remind you to take a look at where are we going to find the Meissner's plexus. You want to look at the submucosa. And that's going to help us, that internal neuro network help with digestive juice secretion, whereas where we find the myenteric plexus, which helps with peristalsis or the propulsion of food. And that's going to be the myenteric plexus, myenteric, myo, muscle. It's going to be in your muscularis layer. All right, there's my references. A lot of these images came from Duke University's histology, which is an open resource if you guys want to use that to study your histo. And then the other slides from your previous tutoring sessions. So with that, I'll take any questions. And if we don't have any questions, that's okay. We can go to the next. Yes, all of these questions I pulled from the learning objectives. Exactly. Any other questions? Okay, thank you all. Oh. On the first slide, why was the epithelium different? Let me see here. Okay. Why is the epithelium different? Do you mean here? Just in general, there was a flaky layer of dead cells, but then there was a purple layer under.
You get the keratinized.
So you have the keratinized layer. And then you have these cells. So I'll go to my next image. So this image, I just tried to zero in here. It's kind of zoomed in. So you can see that this is dead skin layer. And you know that because I can't see any nuclei or those really small dark staining dots. If you look a little closely here, you can see the nuclei. And this is just to show you that they're layered on top of one another. I will say that some of these, depending on the staining that occurs, your skin layer can show up differently. I actually think too, I think Gabriel did a lecture recently talking about depending on if it's dark skin versus light skin, this area will stain darker if it's dark skin. So and that could also be why it's staining that dark.
That's exactly right, Dr. Haggerty. Yeah. This was a slide he used that showed darker pigmented skin from a different individual that had lighter pigmentation. So yeah. Excellent.
And you'll see that. And that's good. It's a great representation to see those changes. Yeah. Or differences. So identifying images. First step is take a breath, because I think a lot of us try to go in and we zero in on the arrow. We're like, what is that? What is that? What is that? So anatomy is all about relationships. That's a big thing, even in histology or anatomy in general. So with just looking at, and I honestly, I didn't realize this until I was even really teaching a whole lot. Because I focused so much on that arrow, and they would say, what's this epithelium? And I didn't even realize, oh, look at that. This is all dense irregular connective tissue underneath here. So when you're looking at an image, just I would take a look. I think with an image like this, look at the whole image itself. Here's my box maybe, or let's go back. Let's go to this one here. So my indicator is here, but what's above it? What's below it? If I look above, I notice that this is a very common thing with keratinized, like there's not a whole lot that looks like this as far as like epithelium goes. So this tells me right away I'm looking at skin, most likely. And then the layered epithelium, looking underneath it, and I see that, and recognizing I'm looking at epithelium here, it's I got my dense irregular connective tissue. This is also recognizing patterns, and this is something that gets caught on after you've studied it over and over and over and over again. So if you're not catching on right away, I would give yourself a break. But trying to just pull up another image here. Something like this is kind of recognizing tissue type. If I'm trying to know if this is a Haversian canal versus a Volkmann canal, I look at other areas of the slide to see if I'm seeing things that are similar. That sometimes gives you a cue. And then I'm trying to think of other things. Do you guys have any other ways to kind of like show too? Color, I don't like to say color because some people are colorblind, and color with histology is very hard to tell the differences. So I always try to, when I'm studying this, always try to grab onto a descriptor when I'm studying that certain type of thing, if that makes sense. Like the epithelium, oh, this has many layers. I don't say, oh, it looks darker because it has many layers. I say, oh, it has many layers. Sorry, go ahead.
The other thing that we should add is to make sure that all the students know that all of the images that you're going to see in our review tonight, as well as all of the images on your exam, are going to come directly from the atlases that we have posted up on our website. So that takes a lot of the guesswork out of it. Like, is there going to be a slightly different image that they pulled off of Google or from some obscure textbook? If you can recognize those images in those atlases, and I think in the histology atlas that the creators did a really good job of trying to put every image next to a black and white line drawing to help you focus more on, as Dr. Haggerty was saying, different kinds of landmarks that you could recognize before you go to the colored image. So try to look at those images, the line drawings, as well as the stained image, and focus on shapes, layers, and recognizable landmarks in these different tissue types.
So I think actually just one disclaimer onto that. I think there might be a couple of that might be from an external source, just because there were a few images that were a little bit more clear, just from a different rotational standpoint. But we promise that there'll be very clear images and that you'll have a sort of a spot that will kind of narrow you down to a couple little areas in there. So, and I will say the majority of at least the ones that I know that I wrote came from the atlas. So that is, I think, predominantly from there.
I did. So yeah, and I did help. I will add to that with putting these on to the histology questions together. It was a little difficult to use the images in the review, only because, as you notice, things can be zoomed out, or you can go zoom in very, very closely. And with the images that are attached, if I would take them and zoom in, it's hard. So I would try to find, from the atlas that I used tonight, I would try to find very similar images from that atlas to what was shown. And if I chose the completely new image, it was because it was very clear what was being shown, and it was because of the relationships that we teach you in these materials.
Thank you. I see a really good question about embryology, but we are going to save that for when we address embryology because we could talk all night about anatomy,
Yes.
histology, and embryology, but we're going to get on to our next session. Um, so with that, Dr.
Oh.
Haggerty, can you stop sharing your screen? And I will go ahead and share mine and we'll do some anatomy. And thank you, Dr. Haggerty, for such great review questions. Thank you to Dr. Frank for helping run our polling system. And thank you to all of you for, just being so great and prepared. This seems like everyone is doing a great job. I was just going to say, if you're not getting some questions, don't be too afraid. Just the fact that you are here, I think, is amazing. And there's a lot, I think, to be said about you just applying somewhere and saying, hey, this is a competition that I was in. So just the fact that you're spending your Sunday doing this again, I think is really awesome. So, all right, we're going to jump over to some anatomy. And I think some of these questions, I think histology just in general and embryology are really complex topics. Hopefully some of this anatomy is a little bit more straightforward or intuitive for you, and we can make up some time there. So with that, I'm going to get to our first question. Got a learning objectives. All right, so first one here. We want to know which organ system is going to be responsible for supporting the body, protecting internal organs, and providing a framework for muscles to attach to. And we're getting a lot of people really knocking this one out of the park. All right, so we had a lot of good responses here for the skeletal system. And so your bones are providing a nice solid framework for you to just have structures to attach onto for your muscles to do that. They're also thinking about your ribs kind of forming a cage around your internal organs, all of your viscera that's very sensitive, sort of tucked more internally. Your skull is in the, housing your brain. So your cranium very nicely protecting your brain and more sensitive nervous structures in there. And so just kind of giving you, if we didn't have our skeletal system, we wouldn't be able to do pretty much anything that we're capable of doing. So very necessary. So circulatory system, we're going to be, getting oxygenation to our tissues. Endocrine system is going to be more of our hormonal sort of function. Nervous system is going to be our electrical impulses to help control the things that we've got going on. And respiratory system is going to be actually that gas exchange occurring for our circulatory system to be effective. So, all right, great job, everybody. All right, so that's our skeletal system, and you can just see here kind of in general how we mentioned our bones. We've got joints again, so these complex sort of attachments that can occur so that we can have our movement there, and then cartilage as well. All right, next question. Let's close this one. All right, so the dorsal body cavity contains which major organs? This takes a knowledge of what does dorsal mean? We've got quite a bit of responses here. Go ahead and share those. All right, so dorsal is going to be more of your posterior cavity, which we can see in this image here. So dorsal is going to be more behind. So that will also include your cranial cavity and your vertebral cavity here. So we could see brain and spinal cord in here. In terms of the heart and lungs, this is going to be found in your thoracic cavity or your thorax. The kidneys and bladder and liver and gallbladder can be found, as well as your stomach and intestines, all in that abdominopelvic cavity that you can see here. All right, and so pretty much everything that's not the cranial cavity and vertebral cavity can all collectively be grouped into be calling the ventral body cavity, also called the anterior body cavity. All right, you guys did a really good job with that one. Okay, next question. So we want to know which of the following vertebrae is being indicated by the arrow. You guys really are doing a really great job with participation here. Really appreciate it. All right, so let's share this poll with you. So this is gonna be your lumbar vertebra. So if you guys have heard of that kind of lumbar back pain, that lower back pain, so this is more of in your lumbar region. So you could think of that lower back sort of area being pointed to here. The other one that I'm seeing is sacral. And so your sacral vertebra are actually going to be fused here. And so this is going to be your sacrum. Collectively are these fused vertebra. You can see a coccyx is going to be at the end of that one. And then above your lumbar vertebra are your 12 thoracic vertebra and your 7 cervical just above that. So cervical is going to be more of your neck region. Thoracic, think the thoracic cavity like we just talked about. That'll extend down, into this abdominal cavity as well, though. And then you'll have your lumbar region sort of in this lower back area.
Dr. Priscura, we had a
Yeah.
question in the chat that I think if you would just take maybe 1 minute to provide the definition of what retroperitoneal means, just so there's no confusion. I think it would take Dr. Frank and I, Dr. Haggerty, longer to type an answer in than for you just maybe to give a definition out loud.
I guess I'm confused. Is the bladder in the retroperitoneal? I don't know if I have a diagram on those. Let's see. So for let's see, I don't think I have a one in there. So the peritoneum is going to be this layer that's sort of encapsulating this space. And so retroperitoneal is referring to the way that this is essentially collapsing onto these structures in there. I'm not sure, Dana, if you have a better way to describe that without getting too complex.
Right. So the peritoneum, I think it was maybe Kathy that asked this question. The peritoneum is just a lining of the abdominal cavity. And so anything that is retro is found deep to that lining. So that would be lying almost on the surface of the vertebral column. And then the pelvic cavity really is not going to be associated with the peritoneum. So retroperitoneal really doesn't apply to any of the organs in the pelvic cavity, and that would include the bladder. Maybe that's where we just leave that for now.
Thank you. I hope that answers your question. That's kind of a complex, I think I know a lot of our medical students struggle with that, but kind of the difference between intraperitoneal versus retroperitoneal, I agree, I think is more of a complex topic, but I'm glad that you're thinking about those things. All right. Our next question here is, what is the role of synergist muscles in muscle function? You guys should have a poll going up here. I know it's a lot to read through. Alrighty, so let's share this one. This is kind of some unique stuff. We got quite a bit of different answers. The main one was actually the correct answer that I'm seeing here. But I see that we also had quite a bit of A and B still in this mix. So when you're thinking syn, that sort of prefix is going to mean same, and then the urgis is telling you that it's producing an action that's essentially is the same. But you have that synergy telling you that it's going to help produce whatever that action is. So there's a different term that we were using to describe B when it was going to be directly producing the movement. Can you guys type in the chat what that was? A prime mover. Awesome. That's great. Yeah, so a prime mover is going to be what is directly producing that movement. So the one that we talked about was going to be the biceps brachii. In this situation could be the prime mover depending on what you're doing. And then the brachialis could be a synergist specifically to this action here. And then we also talked about muscles that are directly opposing. So if you guys could type in the chat, what's the name for something that directly opposes an agonist. Antagonist. Yeah, so adding that ant onto that is going to tell you that it's doing the opposite of that one. So knowing the sort of these prefixes and suffixes can really help you guys out with knowing kind of determining the function. So I know this is a lot to read through, especially if you feel, compelled to be like, oh, we got to rush through this question. Then I'll just say on the exam, just try to think bigger picture, broader, and take a step back whenever you guys are confused by these. All right. Oop. So which of the following muscles abducts the shoulder joint when it contracts? If you didn't see what that previous slide that I jumped to, if you guys can go ahead and answer that one. Take one more minute. Right, so let's share this one with you guys. So a lot of you guys, either you knew it or you saw it because I jumped the gun there. So this is going to be your deltoid muscle here. And so the other two that really came up commonly, one of these is going to be biceps femoris muscle. So that one's kind of a tricky one. So you were probably thinking of biceps brachii when you were thinking of that, because you knew that maybe it was part of the arm. So here, knowing kind of these Latin roots is really going to help you too. So biceps femoris, if you're thinking of femoris, is actually going to tell you that it's part of the femur, so it's going to be in the leg. So that one will tell you, if we're talking about the shoulder action, then you can go ahead and just throw that option out. The deltoid is going to be the correct option. We can see that it sort of forms a cap on your shoulder. So if that's going to contract, you can see how it would lift the shoulder joint up and then abduct it or take it away from the body. Pectoralis major is actually going to be an adductor, and you can think of how that one is going to at the shoulder joint based on its attachment point, more on the chest here, how if that contracts, how that's going to pull in. And if you guys can flex your pecs, then you guys would be able to feel that it sort of pulls your arm in while you're doing it. Sartorius, we didn't really get a lot, and you guys were good with that one. That's a lower limb muscle as well. And then triceps brachii is another one, which we can see that one labeled here, is going to actually be more of, with this long head here, is going to be another adductor. So if you could see if this would contract, if that muscle shortens, how that's going to bring the limb closer to the body rather than away from it like the deltoid will. So when you guys are learning all these muscles and you're doing these actions, just feel what muscles are being activated on. You pull your arm up and you could feel that your deltoid's a little bit tighter, and then put your arm in really close to your body and you'll feel that your triceps are actually a little bit tighter. So you can use your body sort of as a cheat sheet when you guys are studying, and then also when you guys are taking the exam for that matter. So hopefully that can help you guys out. I just put one more image in here in terms of how you can see that this deltoid is forming a cap over the shoulder here, and how that's going to, when contracted, help abduct or pull it away. All right. This one is I think, a tougher question, but I'm really excited to see how you guys do. So in what anatomical structure of the nervous system would you find collections of specifically sensory neuronal cell bodies? I think I accidentally minimized my polling window. Whenever that's done, if you could share that, Dr. Frank.
Okay. Still a few coming in. Okay. 3, 2, 1. Can you see that now?
It's still not letting me. How'd we do with this question? Oh, there we go. Okay, I can see. Oh my gosh, you guys, I'm thoroughly impressed. I could not have answered a question like this in high school. I'm very impressed. That is awesome. And the answer that I'm seeing that a lot of other people said was gray matter, and there's a very reasonable explanation in terms of why you guys thought that. So I just wanted to kind of share the anatomy of that because I knew that this was a more complex topic. So when we're talking about afferent or our sensory neurons, we're taking this information from the periphery and we're going to bring it back to the central nervous system. So let's imagine we've got a neuron coming through there. We said that the cell body is then found in the dorsal root ganglion, again, that posterior root. and then it brings it back through here, and then we're gonna synapse with a neuron in the spinal cord. And that is also going to be in your gray matter here. So I think that kind of is a, if you're talking first order, second order, then both of those options would be correct. If we're talking about efferent and how that's coming out, these are the ones where these more peripheral or these lower motor neurons, these ones are going to be cell bodies directly in that gray matter. And so these are the ones that you're going to see from this ventral horn, and these are going to come out. If it's autonomics, it's going to synapse with a ganglion out in the periphery. And then if it's going to one of our muscles, then we're going to just have that one come out through this ventral root and then out to the muscle. So you guys did such an excellent job with that, and kind of knowing the difference with this ganglion, and then also indicating that there are some of these cell bodies in the gray matter. Great job with that. All right. So now we want to know which of the following best describes the functionality of the central nervous system. Got a lot of good ones for this one. Okay, you guys are doing awesome. All right, so you guys did really good in identifying. This is going to be predominantly for processing and integrating the information from a lot of these other descriptions here. So in terms of what's bringing in information from the extremities or your body, that's going to be your peripheral nervous system. The fight or flight response, you do have autonomic components, working with your central autonomic network, and that's within your central nervous system. But in terms of what the overall response is, this is more getting at your sympathetic nervous system. This is a better descriptor for that. For voluntary movement, this is going to be your somatic nervous system, and rest and digest is going to be your parasympathetic. So if you were having a hard time kind of navigating all these different parts of the nervous system, this was a really good summary slide that was found in the neuroanatomy tutoring session. So you can come back to this. So basically all this is to say is that the peripheral nervous system's job was to bring all this information back for the central nervous system to, again, integrate, kind of modulate the response of. So I kind of like that this one's hovering in the corner because it does have roles from all of these that it's playing into as well.
Dr. Pescara, could you just kind of define what integrate means?
Yeah, so there's going to be a lot of different synapses or connections within all of these pathways. So you can think about how a pain pathway, for example, I'm going to touch something and it's going to be painful. That's coming in from the periphery, and that's just going to come in and say, this was a painful response. But we also have negative emotions that are associated with that. So the central nervous system will also modulate, hey, this was a negative experience and I didn't like that, and I want to stay safe. The central nervous system also has reflexes playing at the level of the spinal cord so that I'm actually withdrawing my hand away from those things in the first place. So those can be modulated cortical, more higher center learning, or it can happen even at the level of the spinal cord. This central nervous system can come into play. I hope that's one example that helps you guys think about how that might work. Thanks for the clarification, Dr. Frank. All righty, next question. So after oxygenated blood leaves the lungs, which chamber of the heart does it enter? Happening right now as we speak. All right, you guys are really good with this one. So the second most common answer I'm seeing, I again could see how you would get to that one. So the correct answer is going to be to the, I can advance this, left atrium. And so you can see through here, blood was going to come in from our superior vena cava, inferior vena cava. You can see through here, brings it back to your right atrium. This is deoxygenated blood from your body. So I can see how you got to that answer because you're just thinking of the general pathway, but this is deoxygenated blood. That takes it to your right ventricle, which then is going to take it out to these pulmonary arteries, because remember, arteries take you away from the heart. we go to our lungs, we get oxygenated, we come back through these pulmonary veins, and it's these pulmonary veins that bring you back to this left atrium. So this is where when oxygenated blood leaves the lungs, it's doing so through these pulmonary veins, bringing you back to the left atrium. The oxygenated blood then goes into the left ventricle, and then it's pumped out to the rest of the body. You guys did a really good job with that one too. And just knowing that atria come first, I think is really, really awesome. And even if you guys missed that question on the anatomy exam, just knowing that for your undergraduate classes now could go a long way. So it's really good. All right, imaging question here for you. So which arrow is correctly identifying the right atrium of the heart? So we just went through a picture of it, but now it makes it a little bit harder because we're looking at a cadaveric image. He's doing really good with that. I love that you guys are doing a good job of anatomical position and knowing that if we're looking at someone else's anatomy, that I'm looking at the anterior view of the heart, then the right is going to be not our right, it's going to be their right. And so we're actually looking at the left side on this image here. That is their anatomical right here. So knowing what the right atrium is, we can see the superior vena cava here is opened up. So that's going to be this structure right here. You can see our coronary vessels sort of separating the spot here, where we could then get into the right ventricle. And then the left ventricle, very large structure, again, on this side. And then here, do you guys know what structure this is? Do you guys want to type that one in the chat, what C is? Yeah, pulmonary trunk. Yeah, that's very good. That's a really complex, I think, question. That's a very advanced one. So for you to be able to call that out is awesome. So that's going to be sitting more to the left than the aorta, which you could see right here. And we already called out the superior vena cava. You can sort of see these pulmonary vessels coming out the back here. But I see in the chat that someone called this out, is that looking for the pulmonary trunk in the aorta is a really good way to identify that you're in an anterior view of the heart versus looking at the posterior. You should be able to see a more flattened structure here, and then you should be able to see your pulmonary veins coming out on either side from there. It's
Yeah,
a
and
really good way to.
I think I like somebody else was saying they saw the coronary arteries branching from the aorta. So if you can see coronary arteries like perfectly branching out and the aorta, yeah, I agree, Dr. Pesquera, that's the best way to see this is in an anterior view.
Yeah, these are very nice and pretty dissected on these, and these came from your dissector atlas, I believe. So that's very helpful. All right, so good job. That was A. You can see the same image here. That should be able to help you a little bit in terms of sort of picturing more of these diagram images that you guys probably started studying from. And I just wanted to make a point to show you guys how you can translate them over, pull them up as a reference when you guys are looking at these cadaveric images. I think that's a really helpful strategy to do. All right, got a next one. We've got, what is the function of the pulmonary arteries in the respiratory system? So a lot of anatomy is knowing what's the function. How does it keep our body going? All righty. I'm going to end this poll. I see we've got a lot of oxygenation of the blood happening with here too. And I could see how you'd think that with us having an artery here. But let me go back to, we can go forward, I guess, and have this slide here. And so this will just show you how we've got our pulmonary artery. So we, again, came deoxygenated blood into the right atrium here. That blood goes to the right ventricle, and then we're going to come out through this pulmonary trunk here, and then we're going to go out into these pulmonary arteries on the left and on the right side, which you can see here. So in terms of what these arteries are doing, it's deoxygenated blood that we are transporting or just taking it to the lungs. So all we have to do here is just take it to the next location. But where, if you guys are thinking oxygenation of blood, what might be instead of pulmonary arteries here, what term might you see here that would better fit C being an answer? If you guys want to type that in the chat. I see capillaries. I really like capillaries is a great answer. That's, I think, just as great of one as you could see. Yeah, so capillaries, where that gas exchange is actually occurring, has to happen in those capillaries, not in an artery and not in these pulmonary veins either. All right. Okay, we've got another one for you. So if you guys could identify the structure indicated by the arrow here, another imaging question for you. You guys are doing such a great job with these cadaveric imaging questions. I'm very impressed by this. All right, this seems to be going really, really well. All right, we'll close that one off. All right, so this is going to be your trachea. So we can see that in here. So aorta is going to be a structure that we just saw in the heart. And so that's going to be sitting more anterior to where we are here. So also diaphragm. What's the diaphragm? Can you guys put in the chat? Why wouldn't it be the diaphragm? It's a muscle. Yeah, this doesn't look really muscular at all here. So what is the trachea instead rather than muscle? It's huge. Yeah. Yeah, it's cartilage. Yeah. Yeah, that's a really good way to describe it. Just noticing these little things in terms of size is, I think, really good things for you guys to pick up on. Esophagus is going to be running very close in proximity. So I'm glad that you guys were able to correctly identify that this is lung tissue that we're going to down here. So this definitely has to be the trachea going into the lungs. All right, awesome job with that. All right, we can see kind of that pathway here now. So you can see how from our pharynx, we've got our nasopharynx, oropharynx, laryngopharynx here, your larynx, then your trachea, you can see right here, and again, how that's going to break off into your main bronchi, into your lungs. All right. Which the following structures contributes to both the foregut and the midgut? This is actually tying in some of your embryological knowledge as well, but still really important because even in the adult structures, we very often refer to the midgut, the foregut, the hindgut when we're talking about these structures because of the blood supply that they're getting, and sort of the folding of the foregut makes a lot of sense then it explains the adult structure. All right, so the one that I'm seeing the most here, other than the small intestine, which is the correct answer. So great job, guys. So the stomach then I could see is the other one here. And so the stomach is actually gonna be part of your foregut. And so then it's actually gonna be part of your duodenum, your jejunum, and your ileum are all gonna be part of your midgut. And then the duodenum is gonna be included in the foregut, the first part of it, the proximal part. And then the stomach, the liver, pancreas, all that's going to be foregut. So I've got a nice summary slide for you guys here. So you guys can see that's going to be a part of our duodenum, and everything else is going to be, that's all of the specific parts of our small intestine. And you can see that part of the duodenum was part of the foregut. Right, so that's what that question was getting at. And then I just wanted to put this slide up just in general to make sure that you guys pay attention to this one. Because again, I think this is one that if you know this table in medical anatomy, then you can get really far on a lot of questions. So even at a high school level, if you can just keep this in a very simple, sort of way, we're not going to ask you anything too complex about this, but if you can just generally describe based on the learning objectives, the organs included, the arterial supply, and the general function of each of these, then you can pretty much get through GI very, very easily. So I just wanted to make sure that was out there again. All right, we've got another one then from a neighboring lecture objective. So what is the function, or it says, what is the function of the structure? But I guess you guys just need to identify the structure here. And then we can talk about function verbally. Oh my gosh, you guys are knocking this one out of the park. And I'd love to hear from you guys. If tutoring session was helping you guys get this, if it was your teachers. But you guys are really doing a great job. All right, so you guys correctly identified this is going to be your liver, and there's a nice little image here. So this is similar to what you guys are going to experience in your imaging questions on your exams. This is letting you know where this image is. And so you can see that this is going to be your liver. And so what is the function? If you guys want to put in the chat, what was the function? A couple of things, filtering blood. That's a really, really important one. All right. Filtering blood. So lots of metabolic detoxification. Yeah. Yeah, this blood filtering and metabolism, very key, important functions by the liver. All right. And we've got, I think, 2 more questions. So which structure in the male reproductive system produces sperm and testosterone? What can we point at here where both of these functions are included? All right, you guys are doing a really good job. All right, so this is gonna be your testes. Be really important for that. We've got a slide kind of showing you what's going on here. And so you can see your testes here. And we can see ductus deferens here, also called your vas deferens. So I want to point that one again, as you guys could probably see either one form there. But again, testosterone, really important here. Do you guys remember, let's go to a histology. Do you guys remember what cells are really important for this? Leydig cells, awesome. You guys are doing a really good job of connecting everything together. That's great. All right, and then last question. Sorry, we're breezing through this end. We're getting close to all the time we pretty much had for the anatomy. All right, so true or false, the female urethra is shorter than that of the male urethra. So one of your lecture objectives was asking you to compare the anatomy of the urinary systems between male and female assigned at birth. All right, some of these true or false questions I think could be tricky because then you end up guessing and get a 50-50 shot. All right, so most of you said this is true. This is in fact true, and so we can see this here. So the urethra for male, we're going to be going all the way through. We have 3 different parts that we talked about in the tutoring session. So you have spongy, you can see here. We have a membranous, that's going to be a very transitional portion, which is why it's also called the transitional urethra. And then you have a prostatic urethra. So very long here versus the female urethra, you can see is just this short portion pretty much directly from our bladder here and then out. So the female urethra is much shorter than that of the male. All right, you guys really did excellent. I am so impressed and so glad that you guys are participating with us. That's all the anatomy that I had. So any questions that I can help answer before we jump over to histology? I'm just seeing, covering what gray matter is again. So gray matter is going to be more of the portion that you're seeing in the spinal cord. That's going to be more internally in the brain. It's going to look like it's a little bit more external, but it's basically just histologically, you can see it's just a collection of where there's predominantly cell bodies located. So if it's if we say white matter, if you're thinking of a highway, it's more of when you're on the highway and you're going to the next destination. So it's gonna be more of your axons. And then when you're looking at your gray matter, it's thinking, okay, I'm gonna stop at a gas station. I'm gonna pick up some gas, pick up a soda. So you're basically synapsing with a neuronal cell body there and communicating something else to another neuron. So it's a little bit different in terms of location for the spinal cord versus the brain. See, but cell bodies aren't collected enough to be considered ganglia or nuclei. So these are actually when there's clusters of these cell bodies within the central nervous system, they're called nuclei. And so there's different clusters of that all throughout your brain. And In your spinal cords, these are sort of aggregated within this gray matter there in your horn there, your ventral horn for motor, dorsal horn for sensory. And then for ganglia, it's the same thing. It's still clusters of neuronal cell bodies, but they're now in the periphery. And so they form sort of a bulge that you guys can even see anatomically. And so then those are called the ganglia. And so then I see here, except for the basal ganglia, that's a really complex thing that you know about. The basal ganglia is a misnomer. And so as such, there's a movement for people to try and call them the basal nuclei, which I think helps to just maybe switch the terminology for that. But those are, in fact, cell bodies within the central nervous system. So they should be called the basal nuclei. All right, the floor is yours, Dr. Peterson. Thanks, everybody.
All right, so can you all see my screen and can you all hear me? Okay. All right, so just so that you kind of have a sense of how my PowerPoint is going to go, I gave you the learning objective. Please just don't try to answer the question quite yet. Just listen closely. I want to give you a sort of overview of how many questions we have and give you a sense of how I'm going to explain the correct answers. So there are 18 questions, and I tried to use learning objectives that were really emphasized in the tutoring sessions and that are really high yield. And what that means in medical school is that these are with a high probability, this learning objective and the content related to it will probably be a question on your exam next Saturday. So having said that, Dr. Frank is going to put up the polls and we'll try to get through all of these 18 questions in a pretty short amount of time. All right, here we go. All right, because of the time, it looks like we're another 5 seconds away from ending the poll, so make sure you get your answer in. All right, Dr. Frank, thank you. So all of you can see that we're a little bit all over the map here. I'm going to go ahead and move on so that you all can see the correct answer, and we're going to talk about this very, very quickly. Okay, so the correct answer was answer choice C. Let me talk you through some of the important information in this question, which I agree, this is a really hard question to start with, but it's a really fundamental and important concept in embryology. So she says that her last known menstrual period started on June 1st. And remember, we've said that the menstrual period is typically about a 4 to 5 day in length or duration. And then that leads in the following week at about day 14, ovulation occurs. And within about 24 hours of ovulation, fertilization has to occur. That's if we're calculating this embryonic or fetal age. It begins at fertilization. So if we do the simple math from June 1st to approximately August the 10th, that would be a total of 8 weeks. But we're given more information. So she said that that was her last known menstrual period. So even though ovulation occurred a week and a half after that, what we have to do to calculate the menstrual age is, and that's also known as the gestational age, is we have to add 2 weeks to what the embryonic age is. So the correct answer again would be 10 weeks. So we'll go ahead and move on to the next slide. So lots of words here. So I'm not going to try and talk too much while the questions go up so that you can really stay focused and think about your answers. All right, maybe just three more seconds. Try to get your answer in. Okay, nice job. All right, so again, I know embryology is hard. I know that there's really not embryology content that's typically explained at the high school level. So most of what you're learning is coming from the Anato-Bee and the tutoring session. So let me go ahead and show you the correct answer here. And let's talk a little bit about this. So remember that we just were referring to ovulation, where the egg is released, fertilization by a single spermatozoa, typically happening in this ampular region of the fallopian tube, and the journey taking as mitotic divisions start to happen takes approximately 5 to 6 days before this blastocyst is going to arrive in the uterine cavity. I'm just highlighting again that the fertilized egg is called a zygote. It undergoes mitotic divisions. It becomes a solid mass of cells, which we call the morula, because it resembles a mulberry. And then once it enters the uterine cavity, about a week after it's been ovulated, it will take up fluid, and that's where we get the blastocyst. And what I'm showing you with my cursor now, there's the inner cell mass. And then what we see on the outside is the outer cell mass. It's the inner cell mass that becomes what we call the embryo or the embryoblast. And the outer cell mass, those cells become what we call the trophoblast cells. And the trophoblast cells, and this will become clear in a couple more slides, the trophoblast cells are actually the closest to the uterine tissue where the maternal blood supply is. So they are going to lead to the development of a membrane we call the chorion, and the chorion plus another membrane collectively are going to be referred to as the placenta. So the best answer here is that the inner cell mass will become the embryo, and the outer cell mass will become these supporting extra embryonic means outside of the embryo itself, the extra embryonic tissues, the chorion, and eventually leading or giving rise to the development of the placenta. Okay. I'm just waiting on a few more of you to put in your answer. All right, thank you, Dr. Frank. I appreciate all your help on this. So the first primary germ cell layer to be differentiated during early gastrulation is what was selected by the majority of you, and that is the endoderm. So again, follow my cursor if you will. I just want to show you what we were talking about in just the previous question and the previous slides. Now we can see definitively, here is our developing embryo. We've got it invaginating, or really just helping with enzymatic action to degrade its way into the uterine wall, which is where my cursor is now moving, so that it can access a maternal blood supply very, very early on, because it now consists of so many cells, and we're talking on the order of hundreds of cells, that diffusion just is not going to be able to happen fast enough for nutrients and oxygen to maintain the viability of all of these cells in the embryo's body. So when we see these different colors, I'm just reminding you that gastrulation has occurred. So our bilaminar disc has gone to a trilaminar disc. The blue is always representative of the outermost layer, which is the ectoderm. Red is the universal color for the middlemost layer and the second layer to form in the embryo, which is the mesoderm. but the very first layer to form, always represented by the color yellow, is the endoderm. And then I think from the tutoring session, this was a really nice slide because it very succinctly also gave you a sense of what different kind of tissues or organs we're going to derive from the endoderm versus the mesoderm versus the ectoderm. So y'all did a nice job with that. And we're going to go ahead and move on to the next question. Hey, nice job. So it's so exciting. I know you've heard this already enough tonight, but really exciting to see that so many of you have spent some time studying this material and are really, showing us that you have assimilated this information, which is just a big 10-cent word for saying you learned this pretty good. So again, this graph is just showing you as we look at the formation of blood cells, that's called hemopoiesis. And here are our months for gestation, so that early on, all of our blood cells are being derived from those blood islands on the surface of the yolk sac. Once the liver has been derived from endodermal tissue of the primitive gut tube, it will take over kind of in the middle months of gestation, that function of developing red and white blood cells. But lastly, really right around seven months of gestation and then peaking, you see that the red and white blood cells are going to be formed within the red marrow of most of the long bones in the body. So great job on that question. And we'll go ahead and move on to the next. Okay, so the majority of you did in fact pick the correct answer. The correct answer, I'm sorry. Let's talk about this because the majority of you did not select the correct answer. And this was asking you which neural pore closes first in the development of the neural tube. And you may or may not remember this, but this was the session that Gabriel Costa gave, and he made this nice little diagram all by himself, which I thought was really well done. So again, what we were trying to show is that in the neural tube, we've got the beginning of the closure of the neural tube. So sealing up that neural crest peak on either side of what's called the neural groove. It starts at the beginning and starts in the middle, closing in either direction. That's where the beginning of the closure starts, is right at the center of the neural tube, but it proceeds slightly faster towards the cranial end where the brain is going to be developing. So it's the cranial end that closes first. So that's called the cranial neuropore, and it's the caudal neuropore that typically closes by the end of the 4th week of development. Okay, so probably need to go back and review that information before next Saturday. Okay, we'll go ahead, move on to the next question. Okay, so let's talk about this question a little bit. So it looks like, yeah, there was really no clear winner here, which means that we should, in fact, have a little discussion about this particular question and the mnemonic that I think is very, very helpful. And oops, yikes. And I hope most of you will take a moment to kind of internalize this as well. So the the neural crest cells are really, really important in the development of many different kinds of tissue throughout the embryo's body. So the mnemonic was Motel Casa, and what you can see is that the beginning letter in Motel and then in Casa are the beginning letters of the tissue types or different kinds of cell types that neural crests give rise to. So I think both Dr. Piscura and Dr. Haggerty mentioned this. If it were me on the written test, I would look at something like this and I would try to immediately eliminate answer choices that I knew could not be involved in the correct answer. So one of the first things I would eliminate is fibroblasts because I know they're connective tissue. So they're not going to be involved in anything really that I care about as forming from a neural crest cell. And the same is true for endothelial cells. Again, these are the lining of those blood vessels that Dr. Haggerty talked about. And connective tissue, remember, and the blood vessels are both derived from mesoderm. And what we're talking about is neural crest cells, and they're derived from the neural ectoderm. So odontoblasts, melanocytes, and Schwann cells all end up here in our mnemonic, making answer choice C the correct answer. This one's a little bit easier. Should go a little bit faster. Okay, so the majority of you picking the correct answer, which is in fact B. And I'm just going to take the same approach that Dr. Prescura and Dr. Haggerty did. Oftentimes, with these big words, if you look at the prefixes, they can give you a tremendous amount of information as clues on what the big word means. So in general, the prefix soma means having to do with the body or with the body core. And splancno is a Latin root word that kind of means having to do with viscera, which is a Big Ten word for having to do with organs. So the somatopleur is that division of mesoderm that gives rise to body muscle. And so, of course, that would be by body, I mean body wall or core body muscles. So those are skeletal muscles like pectoralis major, pectoralis minor, intercostal muscles between the ribs. While the muscle tissue derived from the splanchnopore, a second layer of the mesoderm, splancno having to do with viscera or organs, the walls of most hollow organs in the body have a layer or two layers, or sometimes three layers of smooth muscle. And what I wanted to point out is I included a link to a really great claymation video that I think if you watch it, a lot of the early embryology, the formation of the trilaminar disc from the bilaminar disc, that will make so much more sense. So I highly encourage all of you to watch that. Okay, well, we'll move on. Okay, I'm going to go ahead and show you the correct answer and also point out another prefix here of this big 10-cent word, sclerotorm. Sclero means hard. So the sclerotome region of the somites is going to become, answer choice A, the vertebrae and ribs of the trunk. And this was a diagram, I think it was Natalie that gave this lecture in the tutoring sessions. So really just want you to focus on right here. You see the neural tube. Directly beneath it, the little black dot was the notochord. And then we had, based on some signaling from the notochord, we had the development of the paraaxial mesoderm, PAM, the intermediate mesoderm, IM, and then the lateral plate mesoderm. And you see that here. So directly next to the notochord, shown here in these red boxes, is where the somites are going to develop. And then each somite develops these three different regions, the sclerotome, the myotome, and the dermatome. And as you can see here, it is once again the sclerotome that's going to form the vertebrae and the spines of the ribs. Myo, both Dr. Pescura and Dr. Haggerty have already mentioned myo as a prefix having to do with muscles, and then the derma directly related to the dermis. Okay, so y'all did a good job on that. We'll quickly move on to the next question. Okay, this is a little complicated, and so correct answer, which not many of you selected as the correct answer, is answer choice C. So again, this, you may recall, was a lecture that was presented by Emmeline, and this was the cardiovascular development presentation. So she did all of these drawings herself, which I thought was great. So when we're dividing the right atrium from the left atrium. So right atrium from the left atrium. Right here in blue, we have to form a septum. And a septum is, you can just think of it as a membranous wall. So we form a first septum, and it actually has two holes that will form in it. The first hole goes away. The second hole, the one that we care about, is called the ostium secundum, and that's the final hole. And then after that has developed, there's a second wall that forms just to the right of the first septum, and it's this sort of flap that is going to get pressed by contraction of the right atrium, and it will force blood from the right atrium through this little maze-like opening and over into the left atrium. The aorticopulmonary or the spiral septum is going to form much later on, and it's what divides the common outflow tract into the pulmonary trunk and the aorta. And then B, it tells you right here in the answer, interventricular septum, and we're thinking about atria. So hopefully, reading these answer choices will allow you, if you're reading them critically, will allow you to eliminate at least a few of your possible answer choices. Okay, we'll go ahead. We're still on cardiovascular. Let's
Dr. Peterson,
yes.
could you just answer, is ostium interchangeable with foramen?
So in Latin, they don't mean quite the same thing. A foramen in Latin usually means some kind of hole or opening, where ostium is usually in relationship to something that's much more circular, like a mouth, like a round opening. But that's really getting into the fine details of the definition. So if you want to think of foramen and ostium being similar, I guess go ahead. Okay, you want to put up a poll? Excellent. All right, so nice job, everyone. So if you were a little disheartened before, you should be feeling much better about yourself and how much preparation you've done for this. The correct answer was, in fact, ligamentum arteriosum, and we're just showing you that that forms again between the pulmonary trunk right here and the aorta and serves as a shunt of this oxygenated blood coming in and then moving into the pulmonary trunk and then moving directly into the aorta in the fetus. Since we're not sending blood out to the lungs for oxygenation, we want to send this oxygenated blood as quickly as possible to the rest of the tissues of the fetus's body, especially up to the brain. So the ductus arteriosus will become the ligamentum arteriosum. Okay, we're going to move on to the next question. Okay, so let's talk about this one since no one chose A as a correct answer. So I just wanted to make sure that everybody was thinking along the same lines as I was. And I think, was it Marla Joe, I think, that maybe gave this lecture? I can't remember. So again, we gave you a mnemonic. Every pulmonologist can see alveoli to help you remember the order, but it's really important that you understand that what has to happen starting in week 4 is that there's a little bud of tissue that comes from the primitive gut tube. So this is that outpouching that will give rise to the entire respiratory system. So we're forming the trachea, and then we start to see these tracheal buds, which are the beginning of the bronchi. The bronchi are then going to branch more extensively, leading to secondary bronchi. Remember, secondary bronchi go to the lobes of the lungs, and then late in the embryonic period, the secondary bronchi will grow or extend even more extensively through mitosis and form the tertiary bronchi. So primary, secondary, and tertiary bronchi truly do develop in the very first stages of the respiratory system development, and that's called the embryonic period. Okay, let's go ahead and move on. Just couldn't test you enough on this, so here's another one. Okay. So exactly. So if we're giving rise from our trachea into the primary, secondary, and tertiary bronchi in this embryonic period. Then the next smallest diameter of airways are these bronchioles. And so those are going to develop in the next stage, which is called the pseudoglandular period. So this is where we develop those respiratory bronchioles. And just to reiterate what Dr. Haggerty told you, it's the respiratory bronchioles that are derived from the terminal bronchioles, and their name tells you, hello, this is where gas exchange will first start to occur. So that's really important for the histology portion of the exam. Okay, yeah, you guys did great on that. The next two on the GI system, for embryology, we just didn't get into a lot of the details, so the next two questions should go pretty quickly. Okay. So all but one of these. So let's just talk about what these numbers represent. So we've got the esophagus at 1. This is from Zach Zook's presentation. And the number 2 I eliminated because this represents the diaphragm and really has nothing to do with the formation of the GI tract. Then you see 3, which is the stomach. You see 4 is liver. You see 5 is gallbladder. You see over here 8 is spleen. You see 7 is the head of the pancreas, and you see 6 is the duodenum, so the first part of the small intestine. But Zach made a really good point, and he emphasizes, I think, quite a bit in his presentation, that it's only this first part of the small intestine that is considered to be part or a derivative of the foregut. Once you have where that major duodenal papilla is, that's a little opening in the duodenum where pancreatic enzymes and bile are going to be pumped into the duodenum, then you are midgut. So we can't include 6 because we've moved past or beyond that major duodenal papilla. So let's try the next one. I think it will go better. Okay, so you're right. So yay. You can see, and it's not that easy to see actually on this diagram, but Dr. Prescura, I think, used the same image. So we see small intestines here, we see ascending, transverse, and descending colon or large intestine, and we know it's right at the junction between the ileum of the small intestine and this pouch-like sac called the cecum of the transverse colon. You can just see the little appendix there. So we are way beyond the major duodenal papilla if we're in the ileum region of the small intestine. So this is all midgut. I feel like we've already talked about this. I hope this goes well. Alrighty then. So yay that everyone remembered our discussion from the beginning of the embryology slides. Again, outer cell mass is going to give rise to cells that have trophoblast in their name, and it's the these two different cell types that will combine to form the chorion. And then what I said earlier is that the amnion plus the chorion, as a little bit more time goes by, will mature into what we call the placenta. So great job on that. Okay, looks like there was some disparity in answers here. Let me get my cursor going here. So this was from Dr. Gagne's lecture, and he had both of these images, which I thought was really great. So you've got the anatomically correct, and then you've got the cartoon. So we're talking about the development of these tubules associated with the male reproductive system. So tubes and tubules, those come from the Wolffian ducts. So epididymis, vas deferens, and then the glands and their ducts that are going to not only generate but also direct a large portion of the semen into the urethra. And again, over in the red box in the cartoon, the epididymis, the vas deferens, seminal vesicles, and ejaculatory ducts are all derived from the Wolffian ducts. And just so that everyone is clear, the Wolffian ducts are the same as in males, the mesonephric duct, not meta, but meso. Okay, great. All right, great job, everyone. Looks good. So we're talking about the pouches. And again, pouches here are on the innermost surface of this structure called the pharyngeal apparatus. And we have said that that innermost lining of the pharynx, the primitive gut tube, is going to be endoderm. And endoderm gives rise to glands. And specifically, we're talking now in the pharynx region, these are going to be glands associated mostly with the neck and the upper thoracic cavity. So you did a great job on that. Here comes the last question. Wahoo! Okay, so just last thoughts, last question. I know you're tired, and it makes my brain hurt to have to think this much for so long. So again, what we kind of tried to describe is the gray coloring here represents the neural tube, and we're seeing the hindbrain region and these different segments of the hindbrain. Remember, hindbrain is really medulla region, what we're talking about. These were called rhombomeres, and we had neural crest cells from specific rhombomeres that migrated into specific pharyngeal arches. And that was really important because they were going to give rise to nerves associated with these arches, and then the different muscles that are associated with those very same arches. So said in a slightly different way, the neural crest cells that migrate into arch number 1 are going to provide the innervation for muscles in arch number 1, and so on and so on. And again, a pharyngeal arch is comprised of these migrating neural crest cells along with mesoderm. So let me be the first, and I'm sure all of the faculty are going to want to jump on. I just want to say how much I've enjoyed these tutoring sessions, getting a chance to know some of you in real time. I hope you're finding these helpful, and we're just really excited for next Saturday to come, and we just want to wish you all just the best of luck.
Yeah, I second that. Good luck to everyone. This has been a really fun ride. And we're like, it feels like for us forever because we've been creating this for over a year, but it's just gearing up and it's really exciting. So thank you all for being here and participating with us.
Yeah, ditto to everything that was said. And just also wanted to add, I heard someone in the chat put in that someone made a Quizlet that people were finding. And I just also love that when people are putting questions in, people are answering and just helping support each other through this. I think that's what the anatomical community is. And I'm just so glad that you guys are joining it and bringing the same energy. So I'm just so glad you guys are all here, and best of luck.
Yeah, I will say, this is a similar format that we used to do with the med students, and they don't participate as much. So we really appreciate all of your enthusiasm. This is amazing.
All right, with that, I will stop recording and wish you guys all a great night, and we'll see you a week just under a week away on Saturday for the competition. So thanks, everybody. Good night.